Welcome back to the lecture series in animal physiology. So, we are into the 9th week. So, we have covered a significant part of the nervous system, only we are left with the tail piece, which is one of the senses which we haven't covered, that is the eyes, which is probably for human being the most prized sense organ, because we, being Homo sapiens, which is Homo erectus, we kind of stand on 2 feet. So, it is the eye which helps us to survive and helps us to kind of take a complete gaze of the surrounding. Based on that, we make our next moves. Apart from the eyes, there is a tail piece which is called autonomic nervous system, which is sympathetic and the parasympathetic. And that part will be the last class, while we will be dealing with probably last 2 class or last class we will be dealing with. That part of the nervous system purposefully I will not cover unless I cover the cardiac system, which will be followed by this. So, today what we will do, we will start with the eyes. So, if we talk about eye, there are invariably few things which will come in your mind. So, I am assuming that, say for example, you do not know anything about the eye, then too you will be knowing this. You must have older people at home, and you must have heard this, they have a cataract surgery. This is one word you must have heard on all likelihood if you are in India, because it is a pretty prominent surgery which happens to most of our grandparents, even our aging parents. This is very common. Second thing, what you must have heard, that eye donation, like people make will that after their death their eye should be removed to donate, and the kind of feel what we get is like, eye will be removed from the eyeball. I mean, that's the kind of first feel, at least I used to get in my childhood, till I learned exactly what eye donation means. So, these are the 2 words, apart from appreciating what vision offers you, we all are well versed with, right. So, just like any other senses, eye follows the same paradigm, which essentially is that we have a camera in our body. We have 2 cameras. These are the 2 cameras, 1 and 2, left and right. These 2 camera captures the real time image. These real time images are transmitted to the brain via something called an optic nerve. Optic means vision, optics, okay, just like auditory resemble, or which signifies hearing. Similarly, optics. So, optic nerve to the brain, and in the form of action potentials, and in the brain these signals are decoded to form or make us understand what we are seeing. Of course, there is a comparison which happens. Say for example, you have seen a person, this person, so you are viewing me, so I am coming on the screen, so you know who am I. But instead of me, someone else stands here. That image which is not stored in your visual cortex, the area which is processing the information, then you won't be able to match. So, you will have to store a new piece of information about that new individual who will appear here. So, in other words, just like any other senses, so the camera, which is our eye, which relays the message to the brain decodes the message by comparing that image with the existing images in your circuit, and based on that, brain tells you, hey, this is so and so, this is Professor Manik Das who is taking the class, or this is so and so, likewise so on and so forth, this is the Prime Minister, this is the President, this is so and so, this is like that, this is my girlfriend, likewise, whatever. So, it's all about a comparison of patterns. So, now in a camera there are 2-3 things. those who have opened the camera or know a little bit of, even about a camera. So, you have something called an aperture. So, when the light falls, you can control the amount. So, if you have opened, unless you are totally into iPhone cameras or cell phone cameras, during my time we used to open the camera and to see, but that's fine. You can still open any camera, or you can go online and go to Google and check what camera is. So, there is something called an aperture in the front. Okay. So, at this point, those who are recording my video, so they exactly understand there is an aperture. So, the aperture decides how much light is going to pass through, right. Then there is a lens which kind of focuses the light, and behind the lens you have two options. One option is those who are old people, they know you could have a film, photographic film behind it, where the image will be formed, or current generation relies on a CMOS plate where the image is being formed. It's the one and the same thing. It's just the electronic version of it, which is real time. During my childhood or during my youth, it was mostly a film. So once the film is exposed, you have to remove the film, and to wash it, and you get the image. But nowadays, you have a CMOS sensor on the back. That CMOS sensor is kind of capturing the image, transmitting the image. So here the difference comes. So that CMOS sensor senses the image and transmit it, in the case of human being, transmit it to a mainframe computer, which is your brain. The mainframe computer matches the image and tells what is what, right. Again, that whole comparison. So now, in the light of these three words, you have an aperture, you have a lens, and you have a CMOS sensor. Let's define the anatomy of the eye. Okay. So now we are into week 9 and lecture 1, W9 L1. Fine. Now let's draw the anatomy of the eye. Okay, this is how. There are several muscles which are involved. I am not getting into in depth of those muscles, that you can go online and check for your academic interest. There are ciliary muscles which are involved in movement and controlling the movement of these things. So, the front of the eye, which is this membrane, what you see, is called cornea. Okay. This is your lens, and behind the eye is your retina. This whole part is your retina, and there is a dark layer in the back, fairly dark. We will come to that. What is it? Which is almost part of the retina, but not a neuronal cell. It's an epithelial layer which is in the back. So, this is your retina, and here the dark layer. Retinal pigment epithelial cell. And behind this, you see a lot of these blood vessels forming the network like this. Just I am putting the red and the blue just to show the veins and the arteries. Okay. So, now one-to-one correlation, this is cornea, this is what I talked about, the aperture, this is the lens, again this is the lens, this is one-to-one correlation with the camera, right. This retina, which is the CMOS sensor where the image is forming. The retinal pigment epithelial cell, I do not have a direct one-to-one correlation for a camera. Then here you have the optic nerves, going to brain for processing. In other words, there is a one-to-one correlation. Say for example, this camera is attached to a computer or image processing software, image processing and image recognition software. So, the green colors are showing your one-to-one correlation with a smart visual system. We will come to that why I am giving you a one-to-one correlation, because that will be very important while we will be talking about eye prosthesis, right. So, now the way it works is an image out here, say for example, this is the object. So, the image is formed like this, it passes through, these are the apertures and through the lens, whatever bending and everything takes place. So, it forms an image out here. So, this is where the image is forming, and this part is all filled with fluid, and this is called vitreous fluid. Now, when we talk about eye donation, what you have heard is, eye donation essentially does not mean that you have to remove the whole eye. Upon when a person agrees that eye should be donated, either post death or during his lifetime, what essentially the doctors does is, I am just putting a color now. So, this is the part of the cornea, this is the cornea. So, they actually, it's called removal of cornea, and this is what is your eye donation. You won't be able to even figure out if it is a dead person whose eye has been donated, you won't be able to figure out that anything has been removed. It's just a thin layer. The advantage or the success of corneal transplantation is that since cornea has a very limited blood supply, so the tissue rejection by the acceptor from the donor by the acceptor is fairly low. There is hardly any rejection. So, tissue acceptance is faster because it does not have residual immune cell sitting on it. Okay. So, this is what we call as keratoplasty or eye donation or corneal transplantation. So, you need donor corneas, and that is why there are always appeal, demand from the government agencies that if you could kindly agree for donation of the cornea. Second layer is, so this cell layer is, corneal cell layer is transparent, okay. And why it is transparent will come to the next layer. They follow pretty much the same paradigm. I am coming to that. Second layer is a lens, which now I am shading with blue. or Prussian blue, or turquoise blue, sorry. Okay, one second, let me, should not lose track of the, okay, this is your lens. So what is lens? And of course this is also transparent. Have you ever wondered that how come a part of our body is completely transparent? Is it not made up of cell, or is it something else? So before I move into what is lens is all about, the cataract surgery, what you have heard. So I told you that I'll have one-to-one correlation of what you have heard, and based on that I will explain the things, right, instead of me putting something out of the world. So the cataract surgery is essentially the blurring of this lens, or the lens gets completely opaque. So what happens, the light which is supposed to, one second, okay, so the light which is supposed to pass through like this, or like this, or like this, whichever way, that light is being blocked because of these kind of obstruction or kind of blurriness which happens. Those black dots are showing the blurriness. So how the lens become blurred? So I told you the therapy against it. If the lens is blurred, our only option is that to replace the lens with a synthetic lens, and that replacement of lens with synthetic lens is called cataract surgery. Sometimes there are ways using laser beam you can remove these cataract some way or other, but up to a point you can do so. You can remove this blurriness, but most likelihood that doesn't work all the time. So that is a laser based surgery which is being followed. So having said this, you have to understand what lens is all about. Now if you look at the lens, so now we'll talk about the anatomy of the lens. So this is how the lens look like, or something like this. Now it's a three-dimensional oval shaped structure. This is made up of, initially when the lens is formed, your lens is actually opaque in the mother's womb, very beginning. So the cells are arranged in a three dimension like this. So I'm drawing it for you like this. Okay. That is all in a three dimension they are forming. The third dimension I am unable to draw out here, but that I'll leave it up to your imagination. Okay. And I'll add component by component, that will help you. And they are arranged in a very interesting array, something like this. Okay, and each one of these cells have the nucleus, have all the organelles present in them. This is the initial lens which is formed, through which by no means the whole light can pass. Maybe partially it may pass through, but not good enough. And it has all the organelles and everything. So, a complete, if you see an individual cell, it will be like this. This is the individual cell, whole lens. Individual cells in the beginning. Okay. So, individual cells in the beginning is something like this. It has a nucleus, it has mitochondria, it has endoplasmic reticulum, it has sarcoplasmic reticulum, it has Golgi body, and likewise and so on and so forth. So, it has all the component in the beginning. But then these cells undergo a very interesting phase in their life, and this kind of thing happens in few cells of the body. One of the cells in which such thing is happen is called RBC, red blood cell, which is involved in carrying oxygen carrier. This happens in the lens cells and partly on the corneal cells. Okay. What happens is, at a particular time of the development, all of a sudden in these cells there is a sharp rise in one enzyme called 15-lipoxygenase. Lipoxygenase. 15 is a position of the functional group. Okay. 15-lipoxygenase. And prior to that, in the case of lens cells, there is one more genetic expression, higher expression happens, that is prior to that. That is expression of alpha crystalline protein. Okay. After alpha crystalline, if I put the arrow of time like this, after alpha crystalline is a sharp peak of 15-lipoxygenase, and 15-lipoxygenase does something interesting. So, the next class what we will do, we will talk about what 15-lipoxygenase does and how that completely change the fate of these wonderful cells. Thank you.